Okay, in this video we're going to talk about Chapter 6, which is Genetic and Developmental Disorders. Um, we're going to first start out with this idea of congenital disorders, and these congenital disorders are ones which are present at birth. Now, congenital disorders can be both genetic or environmentally caused, and congenital malformations are structural defects due to errors in fetal development, but these are mostly of a genetic cause and sometimes environmental. Typically, these go hand in hand, but sometimes they are differentiated. Now, some inherited genetic disorders can appear later on in life, and although they're heritable and therefore genetic, if they're not present at birth, then these are not called congenital disorders. These are just genetic disorders, and we'll get to some genetic disorders later on in this lecture. Now, one of the first people to notice this idea of heritability was a gentleman named Gregor Mendel, who was a monk, and his hobby was basically growing pea plants and noting their characteristics and then crossbreeding them and seeing what the sort of progeny or the children of those pea plants looked like. And through some very careful documentation, he developed some of the first ideas of predictable inheritance that really form the basis of how we look at modern genetics. Now, what we need to define, though, is this idea between phenotype and genotype. Phenotype are the outward sort of physical characteristics or biochemical attributes of an individual that are based on someone's genes. Now, what's different, though, is that phenotype doesn't always relate to genotype because genotype is someone's unique genetic makeup. But what's interesting, though, is that certain genotypes can give rise to certain phenotypes. So they are similar but different. So when you think phenotype, think outwardly apparent physical or biochemical attributes. And when you think genotype, think of someone's genetic makeup. So if you sequenced someone's genes, that would give you their genotype. However, if you sort of wrote down someone's physical characteristics, that would tell you about their phenotype. And although these go hand in hand, they can be different for a variety of reasons, which I don't think we're going to get into. Now, in terms of the principles of inheritance, ultimately what we're talking about is someone's basically genetic makeup. And it turns out that people have 46 chromosomes. Humans have 46 chromosomes. And these 46 chromosomes come from these two identical linear chromosome units. And they resemble an X, but these chromosome units separate during meiosis, which is the formation of sex cells. Now, the centromere is the point at which the X forms. So when the chromosomes cross, that little center point where they cross, that's the centromere. This is where the sister chromatids join, or the chromosomes. And this is actually one of the anchoring points for cell division. Now, most of the cells in an individual's body are said to be diploid. And when they're diploid, what this means is that human chromosomes occur in pairs, hence the name diploid. So diploid cells would have one chromosome from each parent. Because the sex cells are haploid, they have half of the number of chromosomes. But when those sex cells combine, they form a full set of chromosomes, which makes a diploid organism, which is you. Now, these pairs of chromosomes appear to be identical because, they're the same chromosome. However, they're going to have different alleles or different forms of genes on each chromosome that come from each parent. So they might appear to be similar, but they are genetically different. So this is what a chromosome looks like underneath an electron microscope. And we can see that it appears to have this sort of X characteristic to it. And really within this X, there's actually two chromosomes. We have our 1 and 2 sister chromatids, which cross here at the centrosome. And if you look out in the periphery, all these little strands are actually individual DNA strands with their histone proteins associated so that you actually can see them. Now what happens though is that as the cell prepares for division, all of this chromatin, or basically DNA and histones, will condense into this chromosomal structure like an X, and then that gets pulled on during mitosis or meiosis, or basically just those forms of cell division. What this particular slide is showing are each of the 23 chromosomes in a human, and you can see that they have different banding patterns and they're different lengths. So not all chromosomes are the same size. So what that means is that chromosomes can have a varying number of genes, or basically a varying number of DNA associated with each chromosome. But generally, the chromosomes are arranged in order of their particular length. And you can see that the chromosomes get smaller and smaller and smaller as we go to the last of the autosomes, which is the 22nd chromosome. So chromosomes number 1 through 22 we call autosomes, and those are the non-sex chromosomes. What that means is that between genetic males and females, we all share 22 autosomes. What differs, though, between genetic males and females are the X and Y chromosomes. So if someone has a genetic makeup where they have 22 autosomes, but then one X and one Y chromosome, that's a genetic male. Or if someone has a genetic makeup where they have 22 autosomes, but then an X and an X chromosome, that's a genetic female. And this comes about during the fusion of sex cells during gametogenesis. Now, it turns out that when sex cells divide, what you end up with then are 4 daughter cells that have half of the number of copies of chromosomes. So if you look here, what we find then is that this is sort of the germline cell, or basically the stem cells that give rise to sex cells. And you can see that this germline cell has 2 copies of the same chromosome. So we call that one diploid. But what you find, though, is that as these cells divide, the chromosomes get split. And so what you end up with then in the end product is actually half of the number of normal chromosomes. And we call these haploid. So haploid cells have half of the number of copies of chromosomes, but they still have a full set. So for instance, if this were a sperm cell, this would have the 22 autosomes and 1 X chromosome. Or this might have 22 autosomes and 1 Y chromosome. So basically having all a full set of chromosomes, but half of the number of copies. This differs from mitosis because with mitosis, we start out with a diploid cell, but we end with a diploid cell. So we still have twice the number of copies of chromosomes. So 46 chromosomes total. In mitosis, the daughter cells have 46 total. In meiosis, the daughter cells only have 23. But this makes sense from a standpoint of forming a new organism, because when you have 2 different sex cells combined, one sex cell, like a sperm, will bring in 23 chromosomes. Another egg will bring in the other 23. When those combine, that will make a complete set of 46 chromosomes, where you have 23 types of chromosomes, but 2 copies of each, so 46 total. Now, genotype, like I mentioned, is the result of 23 maternal, or coming from your mother, and 23 paternal chromosomes. Now, the maternal chromosomes are always going to be 1 through 22 autosomes and 1 X chromosome. So the egg will always contribute an X to that individual. What differs, though, is in the paternal lineage, you'll have 22 autosomes, but then the sperm might carry an X, or it might carry a Y chromosome. And depending on the sperm that fertilizes the egg, you'll end up with a genetic male or female. So if the sperm carries 22 autosomes and 1 X chromosome, if that sperm fertilizes the egg, that will create a genetic female, because then that zygote will have 22 autosomes and 2 X chromosomes, which is genetic female. Now, meiosis is this process of cell division, like I mentioned earlier, where from one diploid cell we end up with haploid daughter cells, and each one contributes 23 chromosomes to the zygote. Now, what's interesting, though, is that there's an interesting sort of effect that occurs during meiosis, and we call this crossing over. So when you have identical chromosomes in meiosis, before these chromosomes divide, they get close to each other at the metaphase plate, and kind of right around the time of prophase 1 during meiosis. You can see that the little branches of these chromosomes kind of cross here, and if you look here, they actually will exchange genetic material. So what you end up with then is kind of a shuffling of chromosomes during meiosis. What this allows for then is increased variability and all of the sex cells that are formed, in fact, there's so many potential combinations of chromosomes and sex cells. The chances of there being any two identical sperm is like 1 in a billion or something ridiculous like that, where these chromosomes cross over, basically recombine and kind of shuffle around, which increases the variability or the combinations of chromosomes in each sex cell. Now, what's unfortunate and kind of fortunate is that mutations do occur every time DNA is replicated. Now, the reason why mutations are unfortunate is that they can lead to deleterious effects, which may cause disease. And sometimes mutations can be fortunate because, and under very rare circumstances, mutations may actually increase the survivability of somebody. They may actually make someone stronger in some way. But that's more rare. In fact, mutations most often lead to damaging effects, not effects that enhance an organism. But mutations in humans are pretty rare. It's about 1 in a million base pairs, or 1 in a million nucleotides that are copied end up being some sort of mutation. Now, there are some potential mutagens, though, like getting exposed to radiation, certain chemicals, and viruses can increase the rate of mutation formation and can make someone more susceptible to something like cancer or congenital malformation. Now, something like a break in DNA is easily repaired. However, if it's a double-stranded break, this may result in loss of genetic information at that break point. So if someone has a double-stranded break and where an entire piece of the chromosome breaks free, that may never recombine, and that can lead to some genetic problems. Now, what this looks like then is like if you have a typical like sort of single-stranded break, what we find is that the double helix is still intact. It's just that one of the strands has a break. And guess what? There's actually enzymes that will come in here called DNA polymerase, and they'll come in here and actually fix that particular break. So although DNA is a pretty stable molecule, it can break apart, but there are enzymes in our nucleus that can fix some of these breaks. The single-stranded breaks are less damaging than something like a double-stranded break. Because if this were a double-stranded break, this end would be broken too, and this entire segment could float away from the chromosome and maybe never recombine. So that can lead to loss of genes in this individual. Now, there's different types of mutations we'll talk about. We have silent, missense, nonsense, and frame shift mutations. If we look here at the slide, what we find is in kind of purplish blue, this is showing the genetic sequence of your DNA strand. And remember, the genetic sequences come in triplets, or every 3 base pairs, you have a codon. So that ATG is the codon for this particular amino acid, or GCA is the codon for this particular amino acid. And so this specific genetic sequence, when it's looked at in rows of 3, we have a specific sequence of amino acids. But this is important to note because if someone gets an alteration to this particular sequence, that can lead to a mutation that may change the sequence of amino acids, or not change the sequence. So an example of one type of mutation is called a silent mutation. The reason why it's a silent mutation is that it's a type of mutation that actually does not change the type of amino acid that's encoded. So if you look here in this particular column, what we're looking at here is basically with this particular mutation, an adenine was mutated into a guanine. However, both TTA and TTG are the codons for the same amino acid, which is leucine. So although you changed one nucleotide, it still led to the same amino acid because sometimes multiple codons can code for the same amino acid. So we call this a silent mutation because although it is a mutation, it didn't change the protein. So it's the same exact protein, just comes from a different genetic sequence, which is kind of odd. Now this differs from something like a missense mutation because a missense mutation occurs when you change a nucleotide, but that also changes the type of amino acid that's encoded by that codon. So what we find then is that maybe before we had TTG, which encoded for leucine, well, if we change that to TCA, that encodes for a completely different amino acid like serine, and that can lead to a missense mutation, which can lead to actually something that's measurable like sickle cell disease. So sickle cell anemia actually is a disease that comes from one point mutation at a specific area in the gene which encodes for hemoglobin. That's all it takes to get that disease, was actually changing one nucleotide. You get a new amino acid, but it changes the protein enough, which causes a disease. Now, that's not so bad when you compare it to something like a nonsense mutation. A nonsense mutation occurs when you accidentally have a mutation that encodes for what's called a stop codon. And the stop codon is basically what tells the ribosome to stop translation. And so it actually truncates or cuts the protein short. So all of the rest of this genetic sequence won't be encoded for. So you find that if we might have before had like a protein with this many amino acids, if you put a stop codon here, the protein cut short, and that can lead to pretty dramatic effects like hemophilia, which is basically a disease where it's more difficult to form blood clots. Now, something else that's kind of odd is there's something called a frame shift mutation, where you might lose an entire nucleotide from the sequence or add an entire nucleotide from the sequence. But remember that these nucleotides are read in series of 3. So if you look here, we find that with this particular frame shift mutation, they've removed an entire nucleotide. So what this leads to then is we actually have shifted the frame, or the reading frame, completely over 1. So it's changed the type of amino acids that are encoded, because you're actually kind of shifting the whole codon sequence over. So what we find then is, although the genetic sequence is similar except for one amino acid, what we end up with then is one nucleotide. What we end up with then is actually a series of different amino acids, because the codons are now changed, because now we actually lost a particular nucleotide. Now, there's lots of different types of chromosomal abnormalities that can occur, but typically if someone survives a chromosomal abnormality, it's usually due to an abnormal number of chromosomes or an alteration of the structure of the chromosome. And this usually results from difficulty with either separating the chromosomes during cell division, or sort of an abnormal shuffling of chromosomes during crossing over, which occurs during prophase 1. Now, one type of chromosomal abnormality we'll talk about is something called aneuploidy. And aneuploidy is what results in an abnormal number of chromosomes. Now, the way you can get abnormal number of chromosomes in a person, or at least a zygote, is either through non-disjunction or what's called anaphase lag. Non-disjunction is the failure for pairs of chromosomes to separate properly during meiosis. So what this means is that some cells can end up with an extra chromosome, and other sex cells will be missing a chromosome. And if those sex cells fertilize the egg, or if the egg is what has those particular aneuploidy, then the zygote that's formed will either have an extra chromosome or will be missing a chromosome. We call this aneuploidy. So you might end up with germ cells that have 22 or 24 chromosomes instead of 23. And this will lead to a combination of a fertilized cell that can lead to 25 or 27 chromosomes. Now, anaphase lag occurs when the nuclear membrane starts to reform. If one chromosome gets left behind out of the nucleus, it will never get back in that nucleus, and it won't be expressed. And basically, it's like missing an entire chromosome. So what this looks like then is that so during meiosis, we find there's a sort of a separation of chromosomes. And if we have non-disjunction, what can occur then is that maybe you end up with an extra chromosome in one sex cell, and another sex cell is missing that chromosome. Or what you might end up with then is that if you get non-disjunction early on in meiosis, you can end up with sex cells that have an extra chromosome and 2 other sex cells that are missing chromosomes. Now, if these sex cells are the ones that get fertilized, then these can lead to aneuploidy. If this sex cell gets fertilized, then this zygote will have an extra chromosome. Or if this sex cell gets fertilized, then this zygote will be missing a chromosome. Now, monosomy is where a daughter cell has a deficiency of one chromosome, and this is not compatible with life, because you can't survive missing an entire type of chromosome. Now, it's not to say that it's missing a copy of a chromosome, monosomy is where you're missing an entire type of chromosomes. Like, for instance, you can't survive without an X chromosome. And that's basically what a monosomy would be. Like if someone were born without an X chromosome, that's not survivable. Or you can't survive without a first chromosome. We call this monosomy. So typically these are not compatible with life, and this is usually what underlies things like spontaneous abortions, where the zygote gets aborted because it won't develop to full term. Now, this differs from polysomy because polysomy has too many chromosomes. So this is going to be like an extra chromosome in the mix. Now, this can result in a viable fetus. However, the fetus will probably have some developmental abnormalities because you end up with what's called gene dosing, where due to an extra copy of that chromosome, certain genes are overexpressed, or you have too many of those genes being expressed, and that leads to developmental problems, and which can affect neurological as well as other organ systems. Now, these are always usually associated with pretty severe disability, but those that are involving sex chromosomes are not as debilitating. So let's say if someone had an extra Y chromosome, that's not as debilitating as if someone had an extra 21st chromosome. And we'll talk about these diseases coming up. Now, some of the causes of these chromosomal abnormalities are pretty poorly understood, but it's believed that things like radiation, viruses, and chemicals may be sort of implicated in these chromosomal abnormalities. However, there are some other risk factors that play a role. So it's believed that advanced maternal and paternal age play a role. So both an older mother and an older father have a higher risk of having offspring with some sort of aneuploidy. And it's believed that these come from abnormalities in sort of the genetic structure of that parent. And the reason why this is believed to be due to abnormalities in genetic structures, it seems to be that some chromosomal abnormalities can run in families. Like Down syndrome, some forms of it seem to run in families. And so it's believed to be due to maybe like abnormalities in the genetic structure of individuals with those chromosomes. Now, the first type of chromosomal abnormality we'll talk about is called trisomy 21. Now, trisomy basically means they have an extra chromosome. So you have 3 copies instead of just 2 copies. And trisomy 21 means you have 3 copies of the 21st chromosome instead of 2 copies of the 21st chromosome. So someone with trisomy 21 is going to have 47 chromosomes, where 3 of those will be the 21st chromosome instead of just a normal 46 number of chromosomes. And we call this Down syndrome. It's due to an extra 21st chromosome. Now, it's one of the most common chromosomal disorders, and it's one of the leading causes of mental retardation. But it also leads to the majority of fetuses that are stillborn or aborted, because many zygotes that have trisomy 21 don't make it to full term. And for those that do, they're usually pretty severely debilitated, and they don't live much past the second decade of life. So some of the signs and symptoms you find with trisomy 21 or Down syndrome are things like mental retardation, epicanthal folds, which are the folds of your sort of upper eyelid, more low-set ears, kind of a more abundant neck skin, an additional crease in the hand, usually congenital heart defects, which also shortens their lifespan, intestinal stenosis, which can usually need to be repaired surgically shortly after birth. For whatever reason, umbilical hernias are common in individuals with Down syndrome, as well as a predisposition to things like leukemia and a larger gap between the first and second toe. Now, this is sort of characteristic of trisomy 21 or Down syndrome. Now, but due to these congenital defects like heart defects, typically Down syndrome people who make it to birth, they won't live like a full life usually. You don't see very many people with Down syndrome like in their 30s or 40s or 50s, because usually they pass away from their congenital defects. Now, some other autosomal chromosome disorders include things like trisomy 18, trisomy 13, and trisomies 8, 9, and 22. Trisomy 18 we call Edwards syndrome. Trisomy 13 we call Patau syndrome. But these are much less common than trisomy 21, but they also cause severe mental retardation. They're not really talked about as much clinically because typically the infants die within the first few weeks of birth because their congenital defects um, are usually not survivable. Now, trisomies 8, 9, and 22 are also extremely rare because if these do occur, usually the fetus is aborted before birth because, again, the genetic defects are not survivable. Okay, so we're going to pick up on different chromosomal disorders. The first of which we'll talk about is Klinefelter syndrome. Klinefelter syndrome is one where a genetic male has an extra X chromosome, and this is going to come from like non-disjunction during meiosis, where one of the sex cells ends up with an extra X chromosome, so that once that egg gets fertilized and you have the zygote, what this means is you'll have 22 autosomes, but then you'll have X, X, Y chromosomes instead of XY for genetic male. So really what Klinefelter syndrome is sort of a genetic male with an extra X chromosome. But remember, the X chromosome is one who's more associated with the female genotype. So if you have a genetic male with an extra X chromosome, it's like gene dosing where they have an additional X, which gives them more female-like characteristics. So what happens is that in these individuals with Klinefelter syndrome, they're usually going to lack secondary sex characteristics. So typically what you think of secondary sex characteristics would be those male characteristics that develop after puberty. Like body hair, growth of certain bones, muscle, voice changes, that kind of stuff. Now, individuals with Klinefelter syndrome will basically lack these secondary sex characteristics. So on this slide is shown an example of Klinefelter syndrome, and this would be genetic male, but XXY genotype. And if you look at this person, they have more of a sort of a female body type. Shoulders are less broad. There's not as much body hair as you can see, and some breast development. So this is called Klinefelter syndrome. Now, another type of sex chromosome disorder is called Turner syndrome. It's present in about 1 in 3,000 births, so it's fairly common. Now, this is a form of a monosomy where it's a genetic female that only has one X chromosome. So we call this XO genotype, where they're going to have 22 autosomes, but only one X chromosome instead of having normally two X chromosomes in females. Individuals with Turner syndrome only have one X. So what happens is that they do have a female phenotype. They're going to look more female. However, ovaries don't properly develop, and what's going to happen is they're going to have more of a short stature, webbed neck. They'll be sterile, have a wider chest. This is usually associated with things like congenital heart defects and lack of menses, or basically amenorrhea. What happens is it's typically associated with advanced father's age. So if the father is a little older, like an older paternal age, they're more likely to have Turner syndrome. And again, Turner syndrome could come about if the sperm never carried a sex chromosome. So let's say if you had a sperm that only had 22 autosomes but didn't carry like an X or a Y chromosome, so that if that sperm fertilized the egg, what you end up with then is the zygote with only one X chromosome, which was supplied by the mother because the father's sperm never brought an X chromosome to the egg. So this is an example of Turner syndrome. You can see that sort of wide chest here, the webbed neck, short stature, and sort of long extremities. And other sex characteristics don't really develop. Like you can see there's not as much sort of body hair here, and they're going to be sterile because the ovaries never develop. Now, there's different ways you can sort of predict the rate at which a certain genetic defect could occur in a family. And we call this a pedigree. So a pedigree is a way we can help trace the transmission of a particular genetic disorder in a family. And based on how we can look at inheritance, we can determine whether or not this genetic disorder is what we call autosomal dominant, autosomal recessive, or sex-linked. Now, remember, autosomes are the first 22 chromosomes. They're non-sex chromosomes. And if it's an autosomal dominant disorder, what this means is you only need to receive one defective copy of the gene in order to get this disorder. Remember, we have two copies of our genes. We call those alleles. But if it's an autosomal dominant disorder, all we need is one defective copy to express the phenotype of a disease. If it's an autosomal recessive disorder, what this means is we actually need to inherit two defective copies, one from each parent, in order to express the phenotype of that disease. Now, there are also sex-linked disorders, which basically are disorders that follow sex chromosomes, like X or Y chromosomes. Usually, these sex disorders are actually going to be X-linked, so they're going to be a gene on the X chromosome, typically because the X chromosome is just larger. Now, what this pedigree looks like is basically just a map. So we can find here would be maybe like the grandparents, and then you have their children as well as their children. So you can look at this sort of pattern of inheritance, and you can see, okay, well, basically the squares are male, the circles are female. If it's shaded in red, that means that that's an affected individual, and by affected means that they actually express that disease. So you can see that in this particular pedigree, most of the people actually don't express that disease, which immediately you can kind of suspect that it's probably not an autosomal dominant disorder, because you'd expect there to be more individuals expressing this disease if it's dominant, because they only need to inherit one defective copy from a parent. You see here that none of the parents are affected, so it can't be dominant. It has to be recessive. So this is probably going to be an autosomal recessive type of inheritance. Now, what this is showing up here is just a more detail on sort of the breeding characteristics that we can talk about in a family, like divorce or illegitimate offspring, relatedness, which is consanguinity, like if brothers and sisters have a child, or if a couple doesn't have offspring. It's just different ways to draw out the pedigree. Now remember, autosomal dominant disorders occur due to a mutation of a particular gene. Now, if it's autosomal dominant, what that means is that for those people who are affected by the disease, they only really need one defective copy in order to express express the disease. What this means is that males and females are both equally affected, and usually it only requires one affected parent. So you might have a parent who's a carrier for this disease, and they might be affected with the disease as well. When they have children, if that parent spreads that defective copy of the gene on their child, then that child will also have the disease. Unaffected individuals do not transmit the disease, because if they're not affected by the disease, which they don't carry the defective copy, so they can't transmit it. Now, in terms of the chances of inheriting an autosomal dominant disease from parents, let's say if you have one affected parent and an unaffected mate, so like let's say if a father has a certain autosomal dominant disease and a mother doesn't, they have a 1 in 2 chance of passing that on to their children. Otherwise, if you have two affected parents, there's about a 3 in 4 chance. So Marfan syndrome is actually an autosomal dominant disorder, and with this particular disorder, it happens to come from a mutation on a gene which is on chromosome number 15. Now remember, it's autosomal dominant, which means you only need to inherit one defective copy of this fibrillin 1 gene from one parent, not even both parents, just it has to be passed to you from only one parent. So in order to get Marfan syndrome, you have to have an affected parent, like a mother or a father that already has Marfan syndrome. And if they happen to pass their copy of the gene that's defective onto you, then you'll have Marfan syndrome. Now, Marfan syndrome is a connective tissue disorder. Typically what happens is that these individuals will be taller, slender, thin arms and legs, thin fingers that are kind of long. We call this arachnodactyly. Remember, arachno means spider, dactyly means fingers. So it literally means spider fingers, but just basically just kind of like longer, slender fingers. Now, this fibrillin 1 gene is actually what encodes for certain types of connective tissues, so that other connective tissues are going to be weaker, like the aorta is going to be kind of weakened due to weaker elastic connective tissue, and there's going to be a lot of cardiovascular problems due to kind of weaker blood vessels in general. So this is an example of Marfan syndrome. You can see that this person is fairly tall, long, slender limbs, and if you look out, their fingers are much longer too, which is that arachnodactyly I was talking about. Another example of an autosomal dominant disease is called Huntington's disease. Huntington's is a neurological disease that it's not really associated with elderly, but the onset of symptoms are going to occur around age 40. So someone could actually have Huntington's disease and not even know it until well after they've had kids. What happens is that we have mental deterioration, which leads to involuntary arm and leg movements. In fact, what happens is with Huntington's disease, people can't prevent themselves from having abnormal movements. In fact, they start moving their limbs around in a way that looks like a dance. So it's called Huntington's chorea, which basically means dance. What happens is that the parent can often transmit this disease to their offspring before they even know that they have it. Now, some other autosomal recessive disorders we're going to talk about coming up next. And what happens is that autosomal recessive disorders come about when you inherit two defective copies from each parent. So, for instance, it's a little bit less likely to get an autosomal recessive disorder because you need two defective copies, one from each parent. So what that requires then is to have two parents that either both of them either are a carrier or are affected by the disease, and if they get together and spread each defective copy of their genes onto you, then that will give you a recessive disorder. So an example of like an autosomal recessive disorder would be like hemophilia. That's a type of bleeding disorder that we'll talk about in later chapters. Now, if you're a carrier, what that means is that the disease is not apparent. So what that means is you can carry a defective copy of a gene, but for autosomal recessive disorders, because if you don't have another defective copy, as long as you have one good copy of a gene and maybe one defective copy, you won't have the disease. In order to express this autosomal recessive disorder, you got to have both defective copies. So what happens is that if you had unaffected individuals that are transmitting these genes onto their offspring, if you have parents that are carriers, they have a 1 in 4 chance of having a child with the disorder. So if both parents are carriers for a certain autosomal recessive disorder, there's about a 25% chance that they will have a kid that is affected by this disease. Otherwise, if you have an affected parent with a non-affected parent, then the chances are about 1 in 2 as having an offspring. So what this is showing is basically something called a Punnett square. And a Punnett square is a way we can kind of calculate the risk of offspring being affected by a particular disease. So the big A means the good copy of the gene. The little a means the bad copy of a gene. So if you look here, we got an affected parent, which means they have the disease, and we can see that here. It's circled in, colored in circle, which means it's an affected mother. Because there's little a's, it means they have two defective copies of the gene. So let's say we have one affected parent who has kids with a carrier of the defective gene. Now, if they're a carrier, they don't express the disease because they still have one good copy, hence the big A. What we can do is set up this little grid here and figure out, okay, well, what's the probability of an affected parent having children with a disease if they mate with a carrier? So we can see here is that, well, we just add these together. So we have, well, one good copy here, one bad copy here. So we have a child that's a carrier here, child that's a carrier here, but then we have two children that are affected. So the chances of having a child with this disease, if you have one affected parent and one parent who's a carrier, is 1 in 2, or basically half. So there's a 50% chance that their children are going to have this disease if one parent is affected, the other parent's a carrier. Now, if we look at this from the standpoint of both parents are carriers, well, we have two good copies in this child, one good copy, one bad copy. But again, remember, you need two bad copies to have the disease. So these two children will be carriers. However, this child would receive both bad copies from each parent. So the chances of two carriers of autosomal recessive disease having a child with a disease is 1 in 4. Now, often these autosomal recessive disorders are associated with relatedness or consanguinity. The reason for this is that if you're more related, the chances are more likely that you are both carriers for a certain autosomal recessive disease, which means if you have a child, then you have even higher probability of that child having your autosomal recessive disease. So there's actually populations worldwide that are more isolated, so people are kind of more related. Some examples are usually island nations. If you live on an island, there's not a lot of opportunity to mate outside of the population on that island. So typically, island populations are more closely related due to lack of migration and immigration. Um, so that you often find these recessive disorders that are common in those communities, or just other isolated communities. Not necessarily on an island, but if they're isolated due to like geographical or social constraints, particularly like the Amish have a lot more recessive disorders because those Amish populations they don't outbreed or anything like that, so they can only inbreed, so they're more often related. So they actually have these recessive disorders are more common. So some examples of autosomal recessive disorders would be things like albinism, which are albinos, phenylketonuria, which is a type of error in metabolism where people can't metabolize the amino acid phenylalanine properly, and so it just accumulates in the body and leads to mental retardation. Another one's called cystic fibrosis, which we'll talk about in later chapters, but it's basically mutation in the gene encoding for a particular chloride transporter, which is necessary for mucus. So with cystic fibrosis, they end up with kind of a thick, sticky, tenacious mucus that makes them more prone to respiratory infections. Now, some other sex-linked disorders include things like certain forms of color blindness, but most sex-linked disorders are going to be X-linked. Now, what this means is that if it's a sex-linked recessive disorder, what this means is it's more unlikely for females to be affected because they need to have two defective copies of the X chromosome. Otherwise, if it's a sex-linked disorder, males are more likely affected because they only have one copy of the X chromosome. So they only need to inherit one bad copy because they don't have a backup X chromosome. So they don't have extra copies of that gene. So the example of this is like color blindness, where there's a lot more colorblind males than females because most forms of color blindness are localized to the X chromosome. So what this means is that males only have one X chromosome, so it's easier for them to have color blindness because they only need to inherit that one defective copy of the gene. Otherwise, it's more difficult for females to be colorblind because they need to inherit both X chromosomes with those defective copies, which is much more rare. Now, for that reason, these sex-linked disorders are almost always male, and so the affected father will transmit a defective gene to all the daughters, and those daughters can transmit that defective gene onto their sons, which inherit their X chromosome from their mother. Now, a carrier female has about a 1 in 2 chance of producing an affected son or carrier daughter, and the reason for this is that because a mother has two X chromosomes, there's a 1 in 2 chance, or basically 50% chance that one of their defective copies will go into the egg that gets fertilized. Now, there are environmentally induced congenital disorders, and so what an environmentally induced congenital disorder is basically environmental factors that lead to maldevelopment of some sort of tissue or organ system. So we talk about the study of these environmentally induced congenital disorders as being teratology, because a teratogen is something that can induce mutations during fetal development. So it's a study of sort of developmental anomalies. Now there's lots of possible teratogens, out there, but typically teratogens can be things like chemicals, radiation exposure, or even viral infections can lead to certain malformations. Like later on the quarter, we'll talk about things like measles and mumps. Those viruses are teratogenic, which basically means that if a pregnant mother becomes infected with measles and mumps, then it's more likely that their fetus will come out with pretty severe congenital deformations. But there are chemicals that can be teratogenic as well. Now, what's interesting though about teratogens is that there's these periods of vulnerability during fetal development. So certain organ systems are more vulnerable to being malformed at different points in development. So that it's not as though exposure to a teratogen is going to cause developmental anomalies all over the body. In fact, exposure to a teratogen in a certain period of time is more likely to affect a certain organ system because there's kind of critical periods of organ system development over the whole 9 month gestational period. So before the third gestational week, so basically before week 3, teratogen exposure either damages very few cells or so many cells that the embryo can't survive. So teratogen exposure early on is either kind of like an all or none sort of process where it usually doesn't damage any cells at all and there's really not an effect, or it damages so many cells that there's going to be spontaneous abortion. Now, between the 3rd and 9th weeks of development, the embryos are even very susceptible to teratogenesis because during this period of time, there's a lot of organ differentiation, and these can be easily disrupted by chemicals, viruses, or radiation. But after the 3rd month of development, what happens is that teratogens will influence the maldevelopment of already formed organs. So there'll be less severe mutations, but they're going to affect already formed organs. So what this particular graph shows are basically some of the critical periods for some of the organ systems to develop. So for instance, between the 3rd and 5th weeks, that's sort of a critical period for your central nervous system. Around the same time, the heart develops in that same critical period. And what comes later then would be things like arms, eyes, legs, teeth, palate, genitals, and ears. And so you can see that there's all these critical windows of teratogen exposure. So let's say if a pregnant mother were exposed to some sort of teratogen, like an excessive amount of radiation, and you knew at what week they were at in terms of their gestational development, you can make pretty good predictions about like whether the fetus's legs would be more affected or whether their central nervous system would be more affected. Now, some different teratogenic agents can be like chemicals or drugs. An example of a drug that's a teratogen is something called thalidomide. And so thalidomide is a drug that was used for morning sickness in pregnant mothers back like in the 50s and 60s. But what happened is that this drug, although it was really effective for the morning sickness, also caused pretty severe mutations during development. And so often the fetus's limbs were malformed, and so they have really short arms and legs. But there are other infectious agents that can be teratogenic. Like I said, measles and mumps can cause some pretty severe malformations, as well as radiation exposure. But sometimes these things can show up later on in life. You know, they're not necessarily going to be visible right at birth. There might be a congenital anomaly that becomes more problematic later on in life, like an aortic aneurysm or something like that. But there are some things that are common amongst most teratogen exposure, and that includes things like microcephaly, which basically means low brain development, kind of underdeveloped eyes, underdeveloped respiratory system, heart defects, enlarged liver, enlarged spleen, as well as underdeveloped blood vessels, which leads to these things called petechia, which are kind of like widespread hemorrhages deep to the skin. Now, what's nice, though, is that there's some technology available now where we can screen for certain genetic defects early on. And some things that you want to consider in terms of screening a pregnant mother would be advanced maternal age as well as advanced paternal age. So a maternal age over 34 puts somebody at risk for certain congenital defects. Otherwise, sort of history of congenital defects is sort of a big risk factor too. So whether there were chromosomal disorders in previous pregnancies or a known family history of certain disorders like X-linked disorders or metabolic disorders or neural tube anomalies in previous pregnancies. Typically, if there's a history of congenital anomalies, that can put someone at even higher risk for future congenital anomalies. Now, some of the methods we can use to screen for genetic defects can include things like ultrasound, where you actually can see the fetus and see how it's developing. Otherwise, we can look at things like amniocentesis and chorionic villus sampling. This is where we can take basically either cells from the fetus or blood from the fetus and look at some genetic markers or serum markers of diseases that may be in that fetus. or we can do embryoscopy, which is more invasive, but it requires sort of surgical endoscopy, where you take a small fiber optic microscope or camera and look at the developing embryo sort of firsthand.